Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the course Enzyme Science and Technology, we are discussing about the different properties of the enzymes. And in this context, if you recall, in the previous lecture, we have discussed about the general properties of the enzymes and what are the historical aspects of the development of this field of enzymology and so on. So in the current lecture, we are going to discuss more about the enzyme classification and how the enzymes are being classified. So the first question comes, why there is a need to classify the enzymes? So what you see here is that we have the thousands of enzymes which are being found and in a single eukaryotic cell, right? Whether it is eukaryotic cell or the prokaryotic cell, you will have the thousands of enzymes, and these enzymes are present in the cytosol, they are present in the different organelles, some of the enzymes are even part of the plasma membranes and so on. So as per the 6th edition published by the International Union of the Biochemistry and Molecular Biology, there are 3196 different enzymes what are present, and I am sure by now the number could be even in some more also. So these enzymes actually catalyze the reactions involving the different types of substrate. For example, you might see that we have the different types of enzymes which are present in the carbohydrate metabolisms, right? For example, in the glycolysis itself, we have the 10 steps, and in these 10 steps, we have the 10 different enzymes which are participating into the catalyzing the different reactions, and they are working with the different types of substrates. And because the enzyme number is very high, because they can be able to utilize the different types of substrates, it is very difficult actually to study these enzyme individually, and that's why it is studying these enzyme individually is not possible, which means if you can actually group them together, it is easy to study them, because then you can actually be able to study one of the representative from the group, and that itself is going to help you to understand the general mechanism. Like, so for that purpose, it is very important that we should do the enzyme classifications. But when people thought of enzyme classifications, the number was very high. They were utilizing the different types of substrates. The question comes, how you can be able to classify the enzymes. So if you recall, or if you might have heard about the classifications of the even the different types of organisms. For example, if you talk about the classifications of the bacteria, classification of viruses, classifications of animals, plants, fungi, or any other groups, they are being classified by taking some criteria. So you actually require some criteria on which you can be able to do the enzyme classifications. In the case of the organisms, it is easy because you can be able to look at the some of the properties and phenotypes and all those kind of thing, and that's how you can be able to adopt that as a criteria, and you can be able to classify them. But in the case of enzyme also, the first question comes, how you are actually going to do a enzyme classification, and what would be the criteria and on which you can be able to do the enzyme classifications. So for enzyme, the as like for the animals or the plants, they can be classified based on whether they are having the four legs, whether they are having the feathers, whether they are actually having the bones and so on. Similarly, for the enzymes, it has only the two properties. One is enzyme, what substrate it is utilizing, and the other is what kind of reaction it is catalyzing. Okay, so enzymes are only doing this, right? So that's why the enzyme classification cannot be based on any other paratype, but also on the basis of only the substrate and the reactions. So that is what it is written, right? So if you want to classify the enzyme can be only classified either by the type of substrate or product what it is utilizing, or type of the reaction it is actually catalyzing. Now if you see the type of substrate or the product, you will see that the same enzyme is actually utilizing the different types of substrate under the different conditions. For example, you have the hexose, right? Uh, so this is a hexose sugar, right? And this can be accepted by the enzyme which is called as hexokinase, right? So hexokinase is also accepting the glucose. Hexokinase is also accepting the fructose. So how you can be able to classify these kind of enzymes which are actually taking up the different types of substrates. That's why the classifying the enzyme based on the substrate is not possible, because the substrate can be changed for a single enzyme. Let's now we have taken an example of the hexokinase, which actually accept the different types of 6-membered sugars, right? Whether it is glucose, fructose, mannose, or any other sugar, hexokinase is actually going to accept these sugars and it will catalyze the reaction. So that's why the idea of classifying the enzyme based on the substrate or the product was turned down, and then the people have started classifying the enzyme whether it can be classified based on the reaction it catalyzes, right? So as for the reaction is catalyzed, hexokinase, whether it will take the same example of hexokinase will actually perform the same kind of reactions, whether it is accepting the glucose as a substrate or the fructose as a substrate. So what it is going to do is, it is actually going to convert the hexose into the hexose 6-phosphate, right? So it is actually going to phosphorylate the sugar, whether it is the glucose or whether it is fructose or whether it is mannose. So that is something which is more and more stable, right, in terms of the reaction it is catalyzing, and that will not change even under the different conditions. Whether the substrate specificity can be modulated for an enzyme, but the reaction it catalyzes cannot be changed until you change the other parameters. So that's how that could be the one of the criteria what based on which the people have started classifying the enzymes. And the union which actually performing these tasks is called as the International Union of Biochemistry and Molecular Biology, which is actually a union of the different biochemists and scientists who are working in this particular field. So they actually have a set of guidelines on based on which they are taking up the different types of reaction what enzyme is catalyzing, and that's how it is actually classifying the different types of enzymes. So enzymes are classified based on the reaction catalyzes by them, okay? And what is the advantage? Why we are doing the enzyme classifications? Because it helps to study the new enzyme, and it helps to study the discovery of the new enzyme as well. So you can imagine that if I have identified the new enzyme, for example, if I have identified an enzyme X, right, and if this X is actually doing this, it is actually taking up a sugar and it is converting that into the sugar phosphate, or it will taking up the DNA and it is converting the DNA into the DNA phosphate. So what we'll say is that this enzyme, which is X, actually which is just identified the new, is having the similar kind of properties what the enzyme kinase group is actually having, or in general what we'll say is it is actually having the properties which is similar to the enzyme belonging to the group transferases, and that's how we can actually be able to say what are the different properties of the kinetic properties or enzymatic properties associated with this particular enzyme. So if it is a transferase, it requires the two substrates so that it can be able to transfer the group from one substrate to another substrate. For example, in this case, it is actually going to use the ATP, and that's how it is actually going to transfer the phosphate from the ATP to the DNA, and that's how it is actually going to form the DNA phosphate. So a lot of informations you can be able to gather about this new enzyme what you have discovered or what you have identified in a organisms or in a particular cell without doing much experiment, because once you know that it is a transferase, it is a kinase, then you can be able to do like similarity search, you can do sort of experiments, and without doing much experiment you can be able to get the basic information about this particular enzyme. Because how you going to deal this enzyme, that all information you are going to get. The specialized information or the specialized feature of that particular enzyme, for that you might have to do the research, and that you will actually going to get in due course of time. But the basic information will help you to understand and to perform the experiments. So as I said, there is a commission which actually, there is a committee which actually classifies the enzyme. So they have actually formed the Enzyme Commission, and the Enzyme Commission has actually classified the enzyme into the 6 different groups based on the reaction what they are being catalyzed. Okay. This reaction mechanism what is catalyzed is not taking into the consideration the amino acid of the sequence, like the protein, the enzyme what you are, it is not taking up into the consideration the protein structures or the chemical modifications. Okay, for example, if you have the enzyme E, and if it is getting phosphorylated, it is actually going to form the enzyme phosphorylated enzyme. So that kind of chemical modifications are not going to taken up into the consideration. Only the native enzyme is actually going to be taken up into the consideration, and all the enzymes are actually going to be classified based on the reaction what is being catalyzed. And all the enzymes are assigned a number which is called as the enzyme commission number, and these enzyme commission number are specific for that particular enzyme what you are going to talk about, right? So that enzyme, when you say, okay, this enzyme is having this enzyme commission number, you are actually going to say that this is the full classifications. So what is the enzyme commission number, and what information you will get from a enzyme commission number, right? So enzyme commission number is a very powerful tool or powerful term which actually going to give you a lot of information about that particular enzyme. So what is enzyme commission number? So enzyme commission number is a number which actually contains the 4 digits. For example, 1.1, 1.1 and 1.1. Okay, so 1, 1, 1 and 1, and it is actually going to be written like this: 1.1.1. And all these 4 1 are corresponding to a particular information, or it is actually going to give you the classification of that particular enzyme. So if I say that all these are A, B, C, D, what is A and what is B, what is C and what is D is corresponding. So here the A, which means the first digit, is corresponding to the class which actually this particular enzyme is belonging. As I said, the Enzyme Commission has classified the enzyme into the 6 classes. So all these are from 1 to 6. So depending on what number you see in place of A, you are going to say that the enzyme is belonging to the enzyme class A, right, or enzyme class 1. Then the number B is corresponding to the subclass under the class, actually subclass, so it will subclass 1. Then the C is corresponding to the subclass, and that also is 1 in this case. And then in the D is actually going to say sub and subclass, and that also is actually the 1. So if you combine these 4, and you will say that this is an enzyme which belongs to the oxidoreductase class, actually, because the first one is the oxidoreductase class. What information the B and C is actually going to give you? The B and C is actually going to give you the information about the reaction what this enzyme is going to catalyze, which means the type of reactions. Whereas D is the final number, and that is the number what is being given particularly based on the actual substrate what is involved in this particular reaction. So you see, from the enzyme commission number, you will get the full information. You will get the information about which class it is belonging, what kind of reaction it is catalyzing, and what substrate. Okay, and that's why this enzyme commission number is a very powerful tool to utilize, and that's how it is actually going to help the scientist to get the full information about that particular enzymes. Now, we'll take an example of some of the enzyme commission number and understand what it means actually. So, EC number are 4-digit number, as we said. Okay, so for example, this is the EC number 3.4.11.4. So what it says is A is equal to 3, B is equal to 4, C is equal to 11 and D is equal to 4. So when it says A is equal to 3, 3 means it is actually corresponding to the class of that particular enzyme. So class of the 3, if in front of the class it is saying 3, then the enzyme is belonging to a group which is called as the hydrolases. If the B is 4, which means it is actually denoting the subgroup, right? So the subgroup 4 within the 3 is belonging to the hydrolases that act on the peptide bond, which means all these are actually the proteases which are actually going to work on the peptide bonds. Then if you have the C, which is 11, which means the subclass is 11, then these are the enzyme like the hydrolases that cleaves off the amino terminal amino acid from a polypeptide. Okay, so that is a kind of the information. So what you see here is that from the B and from the C, we are getting the information about the type of reaction it is catalyzing. First is it is a hydrolases which is act on the peptide bond. The second is that it is actually going to work from the amino terminal side of the enzyme. And then the number 4 is going to give you the information about the subclass, and that is the hydrolases which act onto the tripeptide. So this actually gives you the information about the substrates. Okay, now if you combine all these information, so what it says is that it is a tripeptide, it is an enzyme which works from the amino terminal side, and it is actually a protease. So if you combine all these, you will say that the enzyme is tripeptide aminopeptidase. So peptidase means the enzyme which actually is going to work on the peptide bond. Amino means it is actually going to chew the enzyme, chew the substrate from the amino side, and the tripeptide means it is actually going to work on a tripeptide. It is not going to work on the bigger proteins, it is only going to work on the tripeptides. So based on the enzyme commissions, there are 6 classes. One is called as the oxidoreductase, so that is the EC number 1. Then we have the transferases, that is the EC number 2. Then we have the hydrolases, that is the EC number 3. Then we have the lyases, these are the EC number 4. Then we have the isomerases, and these are the EC number 5. And then we have the ligases, and these are the EC number 6. So let's take the enzymes from each group, and we'll see how these groups are further being classified under the enzyme commissions. So first is oxidoreductase. As I said, it is the EC1. And what are the enzymes what reaction it is going to catalyze? It is actually going to catalyze a oxidoreduction, which means it is actually going to have the transfer of proton, hydrogen or the oxygen, or it is actually going to have the withdrawal of the electron from the one substrate to another substrate. So it is actually going to catalyze the oxidoreductase reactions, which means it is actually going to catalyze the withdrawal or the addition of the electron from the one substance to another substance, which means if you have a substrate AH, if you add the another substrate B, this enzyme is actually going to withdraw the proton or the hydrogen from this, and it is actually going to transfer that onto the B, and that's how it is actually going to form the A plus BH. So this is a reduction reactions. Similarly, you can have the A and it is actually going to add the oxygen, and that's how it is actually going to form the AO, and that's how it is actually going to, this is a oxidation reaction. So basically these enzymes are going to catalyze the oxidation and reduction reaction, and that's why these enzymes are very important in terms of running the metabolism and acquiring the energy for the system. Because when they are actually going to do the oxidation, they are actually going to be a part of the catabolism, and when they are going to do the reduction, they are actually going to be a part of the synthesis phase, because reduction means you have synthesized a new molecule, oxidation means you have destroyed this actually, okay, because there is no A available now, it is the AO what is available. So that is, but in the synthesis part you are actually going to have the energy which is going to be taken up, whereas in the catabolic reactions the energy is going to be produced. Since the electrons are involved in these enzymes reactions, you are always going to use the different types of cofactors. For example, in this case NADH or FADH can be a cofactor. Examples are the dehydrogenases and oxidases. For example, the lactose dehydrogenase, alcohol oxidase, both of these enzymes are part of the anaerobic oxidations. Now, the oxidoreductase class is further being divided based on the subclass and subclass. So, what are the subclasses are present within the oxidoreductase class? So, you have the EC 1.1, which is, these are the enzymes which are acting onto the CH and OH group of the donors. Then we have the EC12, these are the enzymes which are actually acting on the aldehyde or the oxo group of the donor. Then we have EC13, 14, 15, 16. All these are actually going to act on the different types of the substrate like CH substrate, CHNH2 substrate, CHNH2. Sometimes they are also acting on the NADH or NADPH. Then we have the EC 1.7, 1.8, 9, 10, 11. And all these are actually the different types of enzymes which are part of these particular mechanisms, and they are actually acting on the different types of substrates. Then we have the EC12, which is actually acting on the hydrogen as a donor, right? And then EC13, which is acting on the single donor with a incorporation of molecular oxygen. So these are the oxygenases. Then we have the EC15, 16, 17, 18. All these are actually the different classes or different subclasses which is within the oxidoreductase, and they are actually working on the different types of substrates, or they are catalyzing the different types of reactions. Then we have the subclass within the oxidoreductase. So what you see here is the 1.1, which is actually the enzyme which are going to work on the CH-OH group, can still be further having the subgroups like the enzyme which are using the NAD or NADP as an acceptor, or you can have the enzyme which are actually having the cytochrome as an acceptor, or you can have the enzyme which is having the oxygen as an acceptor. So based on the acceptor, it is can be further classified into the subclass, and that actually is going to give you the this EC numbers. Okay. Then we have the 1.2, which is aldehyde, the enzymes which are acting on the aldehydes, or 1.3, 1.4. These are the enzymes which are working on are acting on the alkenes or the amines. Then we have the, and these are the reactions what the oxidoreductase is going to catalyze. So in, for example, in the case of 1.1, it is actually going to work on to the alcohol groups like the RCH2OH, and with the help of the acceptors, it is actually going to convert them into the aldehyde, and that's how it is actually going to have the dehydrogenases. Then we have the 1.2, the enzyme which are actually going to act onto the aldehydes or the carbonyl groups, and they are actually going to convert the aldehyde into the acid, and that's how it is actually a oxidation reaction, right? Because you are converting the aldehyde into the acid, and it is actually going to use the NAD plus as a cofactor. Then we have the enzyme which is going to work on to the alkenes. So in this case, you can have the CH2 and with this another group, and it is going to use the FAD as the molecule, and that's how it is actually going to bring the bring the double bond into this. Okay, and then we have the another molecules like the another molecules like CHNH2 group. So 1.4 is belonging to CH2NH2 group, and that also is going to catalyze the dehydrogenation reactions or oxidative deamination. So these kind of reactions are very common in terms of the some of the detoxification reactions, what we are going to discuss in detail also. Then we have the CNH group, the enzymes which are going to work on CNH group, and that is going to be catalyze this kind of reactions. And then we have the 1.11, which is the where the H2O2 is going to be used as an acceptor. So you see that the H2O2 is going to produce the water molecule and the oxygen, and that is the classical reaction going to be catalyzed by the catalases, or sometimes the H2O2 is going to be used for the oxidation of the substrate, and that is actually going to be a classical reaction what is being catalyzed by the peroxidases. Then we have the 1.13, and that is the incorporation of the oxygen, right? So that is called as the deoxygenases. And then we have the 1.14, which is where the oxygen can be used as an oxidant, and in that case these are actually going to oxidize the molecule, and it is going to be a part of the monooxygenases system. These monooxygenases systems are present within the liver for the detoxification reactions, for catalyzing the detoxification reactions. Okay, so catalase, you see that this is the EC number for catalase, which is 111.1.6, right? Because 111.1.6, and that's how it is a hydrogen peroxide oxidoreductase. Similarly, we can have the peroxidase, which is 1.11.1.7, and there you have the hydrogen peroxide oxidoreductase. Now, when we talk about the oxidoreductase play a very crucial role in terms of the many of the metabolic reactions and many of the metabolic processes. So one of the metabolic processes where the oxidoreductase play a very crucial role is the anaerobic oxidations. So within the anaerobic oxidation, what you have is you are under the oxygen in the presence of oxygen, the pyruvate what is being synthesized by the glycolysis is actually going to be enter into the TCA cycle, and that's how it is actually going to produce the energy. Whereas in the absence of oxygen, the pyruvate is going to be converted into either into the lactate, or it is going to be converted into the ethanol. And this is the pathway where you will see that lot of the oxidoreductase enzymes are involved. So like for example, whether the pyruvate is getting converted into lactate, that is being catalyzed by the lactate dehydrogenase, and you are going to use the NADH as a cofactor. So that is the reaction what is going to be catalyzed by one of the classical oxidoreductase enzyme, that is the lactate dehydrogenase, which is converting the pyruvate into the lactate. And this is a survival pathway, because you cannot have the continuous supply of NAD plus if there is a no oxygen, right? If there is a no availability of oxygen, that there will be a shortage of NAD plus, and that's how this is a kind of a rescue mechanism through which the NAD is going to be generated, and that is actually going to be available for catalyzing the different types of reactions. So when we talk about the anaerobic oxidations, the pyruvate is going to be converted into alcohol in two-step process. The first step, which is going to be catalyzed by the pyruvate decarboxylase, and the second step, which is actually going to be catalyzed by the alcohol dehydrogenases. So this step we are not going to discuss, because this is a step what is being catalyzed by the lyases, right? So that is we are not going to catalyze reaction, what we are not going to discuss at this moment, because that also we are going to discuss when we are going to take up the lyase group. What we are going to discuss is this, right? So acetaldehyde is getting converted into the ethanol, and this reaction is involving the cofactors like the NAD plus and NADH, and that's how what you see here is this is the mechanism through which the alcohol dehydrogenase is converting the acetaldehyde into the alcohol. So what will happen is that the substrate acetaldehyde is actually going to form the enzyme bound zinc. So what you see here is that the zinc is actually also a very important cofactor what is involved into the reaction mechanism. Then it is actually going to bind the NADH, and then there will be a transfer of the hydride ion from the NADH to reduce the acetaldehyde, and once you reduce the acetaldehyde, it is actually going to form the alcohol. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss more aspects related to enzymes. Thank you.